Hello students, welcome back to the channel. In this video, we are going to study how T helper cells are activated and how then they will activate cell mediated immunity and or humoral mediated immunity. You know very well basic till now that two main lymphocytes are there which play very important role in adaptive immunity. One is T lymphocyte, another is B lymphocyte. T lymphocyte is again of two types, T helper and T cytotoxic cells. So first of all, the T helper cells, we have to activate them because T helper cells themselves do not carry out the response just like humoral or cell mediated. So they have to be first activated and then these T helper cells will activate either B cell or cytotoxic cell. Now, what are the two important things to make the T helper cell to activate? First of all, the pathogen which is processed by antigen presenting cell. Okay, few cells are there, specialized cells like dendritic cell, macrophage or B cell. They are known as antigen presenting cell. So they will process and display the fragment of the antigen on their surface. The first, the antigen presenting cell has to display the fragment on their surface, and second is now helper T cell should specifically bind to this with the help of its receptor, antigen receptors which are present on the T cell. Is it clear? Now antigen presenting cell, they are special. Why? Because they are having two type of MHC. Antigen presenting cell will have MHC1 as well as MHC2. Remaining cells nucleated will have only MHC1. Now see next. See, this is a diagram of one antigen presenting cell, dendritic cell. After doing phagocytosis, it is displaying the piece on MHC molecule class 2. Clear? It is displaying the piece on class 2. And now this is T helper cell which is binding to it, the fragment with the help of its receptor. It is binding to this fragment with the help of its receptor, and further lock and key like binding means proper binding is done with the help of CD4. Now CD4, they are special type of protein. They are a cluster of differentiation. CD4 is present on T helper cell and CD8 you will find in T cytotoxic cell. So right now we are working on T helper only. So I'm just focusing on CD4. Now, when the two cell interaction is going on, then what will happen? Cytokines will be released from the dendritic cell. Dendritic cell will release cytokine. It's a type of a chemical. And it will stimulate the T helper to produce its own cytokine. It will also produce its own cytokine. Clear? So cytokine released from APC will stimulate this T helper cell to produce its own cytokines. And when the contact is occurring between them, what will happen? Now T helper cell is activated. Once it is activated, now it will go for cloning to form large number of activated T helper cell. Now, this activated T helper cell can activate cytotoxic T cell to start cell-mediated immunity. Or it can activate the B cell to start humoral immunity. Now, very important, understand one very important concept. B cell, once which is selected, B cell, it is selected. Okay, it is selected by binding to the specific antigen. It can bind to the specific antigen, it is selected, but this B cell will not be activated till the time it is not making a connection with the T helper cell. And that also which T helper cell which is activated. Means selected B cell will bind with T helper which is activated, okay, activated T helper, and then only we will find B cell will be activated. See here, B cell present antigen to already activated helper T cell which will activate the B cell to start humoral immunity. So I think so in this video, we have understood how the T helper cell activation is done. And once this cell is activated, then how it will make the T cytotoxic cell to activate and or either make B cell to activate. I hope so this video might be helpful for you. Thanks for watching.